I'm able to share my screen. So ma'am will be able to share.
No, I've shared the screen.
Yes.
And it is giving me several options. Windows Explorer,
You have
screen
to
one. How
select
do I
your
go to my presentation on my desktop? No, the folder is on the desktop.
open your presentation, ma'am.
Open it.
Presentation. Yes, yes.
Okay, 1 minute.
Ma'am, you have opened your presentation?
Yes, I've opened it, but it's not on full screen. It is because if I do full screen, I cannot come.
No problems. Now you go to
Now I'm going to share.
Go to share.
Yes, I can see. I can see my presentation.
Now you select it, ma'am.
It says ICMR NIRH will stop sharing content as soon as you start sharing.
Okay.
Yeah.
So
So
you
should I share now?
yes, yes. You have to check now whether
Yes.
your
Yeah.
slides are okay or not. Yeah, you are able to do that, right?
Yeah, we can see the
now,
slides. Yes.
Now I'm okay.
Yes. Now you can see the screen. Yeah.
Yes, perfect. You all can. The slides are moving. You all can see them moving.
Yes, we can see the slides and they are moving also. Perfect.
Great, Sujata.
Thanks.
Yeah. Thanks,
Great.
Taruna.
Yeah.
So we'll wait now. No?
Now you can put the first this thing flyer slide. Ma'am, you can stop sharing.
I'll stop sharing.
Yes, ma'am. We will do this process again for you.
Stop sharing. Okay.
Yeah. Thank you.
Good afternoon, everyone.
Good afternoon,
Madam.
Good afternoon.
Hi, good afternoon, Bala. Good afternoon.
So I will just call Dr. Senes because sir has not joined yet.
Yes.
Yeah. Still 10 minutes. So.
Yes.
Yeah,
we have time.
I'll just give him a call. Yeah.
Yeah.
Hello.
Good afternoon, sir. I'm Sujata this side. Thank you so much for joining. Yeah,
Hello, Sujata. How are you?
I'm very fine, sir. Thank you so much.
Hello, Dr. Chiprankar. Dr.
Hello.
Tarana. Nice to see you people after such a long time.
Such a long time. Yes.
Yes.
We had like lot of postponements for ma'am's seminar, and so I kept bothering Senis sir also.
Yeah.
And this always, putting a new date. And thank you for your cooperation, ma'am and sir. We are finally seeing this day. Thank you so much.
Right. It was a very difficult time for me, shifting and all that.
Yes, ma'am. Yes.
Thank you for accommodating.
No, ma'am. It's a pleasure.
It's always a pleasure to chair a talk by Dr. Chiprankar.
Oh my God.
Yeah.
We
I
have
I had
double
a few
pleasure.
occasions, so
Yeah. So we all are having a double pleasure of having you and Professor Chiplankar, I think.
So we will wait for some more time. I've
oh,
been
sure,
sending messages
sure.
for people to join in.
How
But
many people have joined so far?
How many have joined?
we have very few people at this
Yeah,
moment.
it's
Hopefully, people will join in another 5 minutes.
yeah.
So you
I
don't
have
mind if I drink my tea?
Yes,
absolutely.
please go ahead.
Yeah.
I can switch off my video if you want.
No, no.
You can carry on like that, sir. Yeah.
In fact, I talked to Dr. Chiprankar after a very long time today.
Yeah. It was
Yeah.
really nice catching up. Taruna, how long should be the talk? Or she's left?
Ma'am, it can be 40 to 45 minutes.
Huh.
I don't think so there is any issue. But if you want to continue for some more time, that also is okay. We keep it for 1 hour.
1 hour.
And our slot is at least for 2 hours. So
But 45 minutes, you expected 45 minutes to 1 hour?
Yeah, ma'am. I think based on the attention span that people have these days, so I think 45 minutes should be okay. Yeah. We have Dr. Aparanji also here now.
Yeah. Sripad Patil has joined, Ravi has joined. Okay.
Lakshana, Ravi Gutti. Dr. Nibriti also said that she's going to join. Dr. Ganguly wanted to join, ma'am, but he's traveling, so he said he won't be able to join. Yeah. And Dr. Mehra, I think, is giving a talk at some place about S20. So, sir is not available.
Is Sita going to join?
No, I don't think so, sir. उनका कोई मैसेज नहीं आया मुझे. मैसेज नहीं आया.
So I hope you will come back to Bombay, Dr. Chiprankar, after these couple of years in Pune.
Yes, yes.
I'm sure you will miss Bombay.
For sure. Everybody asking us how do you find Pune? How do you find Pune? So I keep telling them I've not seen the real Pune because we have not moved too much, and second we are in Wakad, so it is really outskirts.
Yeah, it's total outskirts. Yes.
Yeah.
In fact, that whole area going towards Hinjewadi and all that, in the evenings, it's so tough getting around there.
Exactly.
Very difficult.
No, but for us, we are very close to the highway. So
Yeah.
actually, Pune Bombay traveling is quite fast.
Yeah, no, traveling from Bombay to there is good.
Huh.
But once you are on the road towards Hinjewadi or even internal,
Right.
That area is now fast becoming institutional area also with symbiosis and all this thing coming up there.
Institutes, oh God.
So how are things at NIRRH, Taruna?
Sir, all good at NIRCH. Ma'am was there day before yesterday. She delivered a lecture in our DHR workshop. Sorry, ma'am, I could not be there because I
Taruna, something you are seen only half. Why is it? Wait 1 minute.
Okay.
Now it's okay.
Now it's okay. So I could not be there on that day because I had to travel to Delhi and just to visit my parents, ma'am.
Who's the new director at NIRRH after Dr. Mahalle?
Yes, sir. Dr. Geetanjali Sachdeva, our colleague, is at now in her place.
Achha.
Yes, sir.
So Dr. Mohanty's ERC, is it still a part of NIV or is it an independent unit?
Yes, good afternoon, sir. Good afternoon, everyone. Yes, NIV Mumbai unit is a part of NIV.
What about your new campus? You were to move to some new place, no?
No, sir. We are there in the Hufkin campus itself.
No, it's still there. Okay.
Yes.
No, there was some, I don't know where it was, some place.
There was one space given to us in Kharghar, but
right.
it has not been developed yet.
Not yet developed.
Yes, good afternoon, ma'am.
Good afternoon.
Just in front of Actrek itself, we have a land which is boundary is there showing NIV Mumbai unit, but for some reason it has not been proceeded further.
In front of Actrek?
Yes.
I see.
Yes.
Okay. But when like it has not been still commissioned or anything, I mean, not given to you all, is it?
No. The land is land acquisition has been done 6 years back. But ICMR is not yet keen to develop it a full-fledged institute because it got merged after that with NIV, and NIV itself is having 2 campuses.
Right.
So still the land is with ICMR. We have a board over there saying NIV Mumbai unit, but it is not advanced for the building or anything.
I see.
Good. Do we have a count somewhere? How many people are there?
Dr. Bala, you wanted to speak to me? You were calling me? Dr. Bala Krishna?
Yeah.
Actually, some of my students are calling me, telling that the link that we sent through email is not opening, it seems. You please check. Some of them, they called.
Okay. So, is it possible for, or should I send another email to everyone? You want me to do that? All the members, because I don't know who are these students.
Yeah, correct. For those individuals, I sent forwarded through WhatsApp. So now it is open. It's opening for them.
Opening?
Yeah. You better you please send to all.
Okay. Okay. But the same link which I have sent it to everyone, sir.
Yeah, that's what I'm wondering. We need to check our members. We have to call and find out.
The link shared by the WhatsApp is perfectly fine. So I have joined through WhatsApp only.
Yes. Correct.
Let us all welcome Dr. Shamal Roy. Sir has joined us.
Hi, Shamal. Nice to see you after so many years.
Sir, you are on mute. Can you please unmute?
Shamal, you need to unmute yourself. एकदम वाइस चांसलर दिख रहा है ये तो. He's really looking like a vice chancellor.
You can see him. I can't.
I can. Can't you?
No, I can't. Wait. I have to move the. Oh yeah, I got a glimpse.
Yeah, Dr. Roy and myself, we used to see each other every day when we were in Boston, and now we see each other once in a blue moon. उसका खाना
So Chinese, how you are doing nowadays?
Good. Quite good. Maintaining reasonably good health. Occasionally getting into academics.
What you are doing? Gardening, traveling?
No. Walking, playing tennis once in a while, giving a talk here and there.
Okay, good.
Socializing.
That's great.
Till about 3 years ago, I used to teach, but now I'm not teaching anymore, so that's okay.
Your kids are abroad, right?
Huh? Teaching immunology in Narasimha University. That was my last assignment.
No. Your kids. Your kids are abroad?
Oh, kids are abroad.
Okay.
He was in Boston till January last year. He has now moved to Portland in Intel, the elder one. Younger one is in Germany for last 12 years.
Okay.
So just the two of us.
Great. Some to Calcutta sometime.
Aiga.
Okay.
How is your son Satyaki doing?
He is joining as an assistant professor at the University of Alabama in mathematics.
Very good.
In August, he will join.
Are you still in that house in?
Oh yeah.
I come to that house. I remember that.
Come over once again with your wife.
Aiga. Oh, you were busy. You were vice chancellor in Kuch Bihar.
No, I'm no longer a vice.
Huh? What are you doing right now? In the sense, currently, where are you affiliated?
Back to IICB.
Oh, yes.
As emeritus.
Very good.
I have seen Shubha because we share in one committee. I have seen her number of times.
Yeah.
We have Dr. Shripad Patil also joining.
Where is he?
Dr.
Shripad, yes, Shripad is also there.
Nibriti ma'am, can you put on your video, and Shripad sir, please?
Where is Shripad?
I can see his.
He's here. I don't see his picture, but he's there.
Welcome, Professor Muthukurappan sir.
Oh, Dr. Muthukurappan is also there.
Yeah.
Oh,
wow.
Great.
Yeah, Arvind, the team of people have joined here.
Oh.
Wonderful.
Hello.
There are 19 participants. Nivriti has joined.
Hello, Dr. Chinese.
Hello, how are you, sir? Nice to see you after so many years.
After so many years, nice to see you. Okay.
It's a real long time. Go back to.
Hemant, we cannot see the flyer slide. Yeah, now. Yeah. So, ma'am and sir, with your permission, shall we start?
Yes.
All a warm welcome to all the members of Indian Immunology Society who have joined us today for the Golden Jubilee online lecture series, which we call it as Pratiraksha Swarna Mahotsav Vyakhan. And today is the 6th lecture and delivered by Professor Shubhada Chitlunkar and would be chaired by Professor Stainis. And I request my colleague of the EC committee, Dr. Sujata Mohanty, to please do the honors. Thank you, Sujata.
Hi, thank you and namaskar and good afternoon to all of you. So it's really a pleasure and my honor to introduce the chairperson of today's scientific session, Professor Krishna Balaji Saini. He is a former director of Biomedical Group, Baba Atomic Research Center, and also a former Raja Ramanna Fellow of BARC. He is the honorary professor of IIT Mumbai. He has been awarded with Shanti Sarup Bhatnagar as well as the INSA Young Scientist Medal awardee.
Sujata,
So I think, yes.
Just a moment. Hemant, can you please show sir
Slide.
slide?
Yes.
Yeah, please. Sorry, Sujata, to interrupt you.
No worries. I think that is very important for the young generation to know about the legacy of our mentors and seniors. Yeah.
Hemant, not this one. We need to show slides for Dr. Stainis.
Sorry, the next batch.
I am traveling and that's why this is happening. I am not there in the institute today.
Anyway, don't bother you. Go ahead.
Just give us a moment, sir. We'll put everything in order. Yes. Thank you.
Yes. Thank you so much.
Please,
yes.
Slides. There are 3 slides. Please move them as Dr. Sujata is going through them. Thank you.
Perfect.
Yeah.
Thank you. So he's born on a very significant date, that is 2nd October. He earned his master's degree in biochemistry from Savitribai Phule Pune University and did 1-year orientation course in biology and radiobiology at BARC. So he joined medical division in 1972 as a scientific officer in BARC. So while working in that same institution, he completed his PhD in biophysics in 1980. He did his postdoctoral studies at New England Medical Center of Tufts University School of Medicine, and with the assistance of IAEA fellowship, he completed his further studies at University College of London at its ICRF Tumor Immunology Unit. So in BARC, he and his team, they have carried out a very significant work in a very diverse area, which is basic immunology, cancer immunobiology, effect of radiation on immune response, immunomodulation by antioxidants and radioprotective agents, constituents of medicinal plant, autoimmunity, and tuberculosis. So he is a great teacher and he taught immunology at postgraduate level in University of Mumbai, IIT Mumbai and Narsi Munji University. And he has really guided PhD students in terms of research methodology and their future course. More than 100 research publication and popular science series article has been his contribution. He is a member of multiple boards, the boards of studies, University of Mumbai, Pune, HDNI and Scientific Advisory Committee of few national reputed institutions. He is a chairperson of Internal Review Board of Tata Memorial and Attract and Institutional Committee for Stem Cell Research. He is also a member of DST Program Advisory Committee on International Collaboration in Life Sciences and Medical Biology. He is a recipient of Homi Bhabha Medal for BARC Training School, Senior Scientist Award of Indian Nuclear Society, Senior Immunologist Oration Award of Indian Immunology Society, really our pleasure, and DAE Group of Achievement Award. He is a fellow of National Academy of Sciences, Nasi, Maharashtra Academy of Sciences. So his list is never ending. He is a former distinguished representative of India at UN Scientific Committee on Effect of Atomic Radiation, and more than 10 years. He is also a former chairperson of International Atomic Energy Agency's prestigious standing advisory group on nuclear applications. So again, as being a director at BARC, Biomedical Group, he supervised and coordinated research activities of 6 different divisions, which includes healthcare to DAE employees in Mumbai and studies on high-level natural radiation in Kerala. Sir, we are indeed grateful for your presence, and I request you to kindly moderate today's scientific session. Thank you so much, sir, for joining.
Thank you very much, Dr. Mohanty. I hope I have unmuted.
Good afternoon, Dr. Sainis.
Good afternoon, everybody. Yes. It's indeed a great pleasure to be back amongst my fellow immunologists, all members of the Indian Immunology Society. And there could not have been a better occasion than the Golden Jubilee lecture series of the IIS. It's also a matter of great privilege as well as pleasure to be chairing this lecture, soon to be delivered by my friend of more than 3 decades and a colleague and a very senior colleague in the institutions of the Department of Atomic Energy, Professor Shubhada Chiplankar.
Hemant, may I request you to please put on the slides for Dr. Chiplankar?
Yeah, I can go ahead. He can put the slide.
Yes, sir, please.
Yes, Dr. Chiplankar has been, her alma mater, of course, has been the former Cancer Research Institute and now the Advanced Center for Research, Training and Education in Treatment and Education in Cancer, ACTREC as it is called. She joined there way back in the early 80s. She did her master's in applied biology from there by research and continued to do her PhD in applied biology under the guidance of Dr. Karande, and late Dr. Karande. Immediately she obtained a postdoctoral fellowship in the laboratory of Professor Stephen Kaufman at the University of the Max Planck Institute of Immunology in Freiburg, Germany. She returned and joined as a regular scientific officer in ACTREC, and then she grew up in her career to become scientific officer H, and then from 2013 for 6 years she became the director of ACTREC. Dr. Chiplankar had varied interests, of course, main primary broad area being the immunological dysfunctions in cancer and development of approaches for immunotherapy. She started her career with murine memory tumor viruses and immune response to them. Then she worked in oral cancers, the antigens of oral cancer, went on to make monoclonal antibodies to things like alpha-fetoprotein, probed various kinds of surface markers of oral cancers. She also did some work on hepatitis C and spent a lot of time working on the ICRC mycobacteria. In Kaufman's lab, she worked on immune responses to mycobacteria, especially the T-cell responses and their regulation. And for some time, she continued on that after coming back from her postdoc. And then somewhere in the 90s, early 90s, I think when Anupama Lad joined you, Shubha, if I'm not wrong,
Yes.
that she switched her attention to this unique set of T cells known as gamma delta T cells. These are typical CD4 negative, CD8 negative cells. And the thing about this, these were gamma delta positive. There is a subpopulation of CD4 negative, CD8 negative cells, which are also alpha beta positive. And these cells could be expanded. She was able to make them grow in culture and then completely probe the immunobiology of these cells. What are the various stimulants for them, how to grow them, then how can they be used, characterization of their T cell receptor usage. And one particular subpopulation that she was keen on, or she worked on, was this V gamma 9 and V delta 2 type cells. So she showed their roles, of course, in different cancers, obtained them from even normal human peripheral blood cells, very small quantity. And then she showed, in recent times, she showed their role in inhibiting the osteoporosis, especially in the inhibiting the resorption of bone by osteoclasts. And they are also, in fact, a dual role, depending on what cytokine it secretes. So that is the kind of work she and her students did. In addition to that, she contributed to work on several other cancers along with her colleagues, and has today more than 100 publications to her credit in 18 book chapters. Above them, she has guided 31 students for PhD. It's a really massive number. When I look at my own number, 18 looks very small. But she has 31 to her credit. She has been on several committees and has been conferred with various honors. Most important among them is at present she is president of the Immuno-Oncology Society of India, a society which I guess was floated about 7-8 years ago, or maybe 10 years ago. And she was one of the architects of that, and now she is president of Immuno-Oncology Society of India. She has contributed to cancer education in a big way. Most important among them is her membership of the IIS Education Committee, to which she is making very significant contribution. Of course, she has been associated with the University of Mumbai, Homi Bhabha National Institute, which is the deemed university of the Department of Atomic Energy, and other universities, as faculty, not just faculty member, but member of the Board of Studies, member of the Academic Council, member of the selection committees for PhD teachers, and so on. So everywhere her mark is there as far as the HBNI is concerned and Mumbai University is concerned. She has been organizer of, she will be the organizer of the PIMSA IIS Advanced Immunology course. And she's looking for one more such course to be organized in Bangalore. She's recipient of several honors, including the oration, that is the Immuno-Oncology Society President's Oration, then Indian Association of Cancer Research President Oration. She was also president of ISCR. So the list is really long. But what's most impressive about it is she was able to interact with so many clinicians, students, pharmacy company people, all these, and integrate their ideas into excellent pieces of work every time published in very good journals. I was very fortunate that I was on the doctoral committee of several of her students. So I came to know about that work, especially the work on gamma delta T cells that she has done on many occasions like that. What's more important,
You should have taken 3 drops.
This to industry. She's got a patent now.
Did not change.
With a person at Bits Pilani, and that is
Keep
on
seeing. That's what you have to see.
Character of breast cancer cells. And now another one has been filed on the therapeutic use of gamma delta T cells. So from bench to bedside, entire range of work that one can think of in cancer research, she has been a party to it. I'm really very proud, Shubha, that we have been associated with each other for such a long time. And Taruna has rightly chosen you to deliver today's Golden Jubilee lecture number 6 for the Indian Immunology Society. May I now invite Dr. Chipunkar to please deliver your talk.
Thank you. Thank you, Dr. Sinus, for such kind words, encouraging words. As he mentioned, we have been colleagues, friends for a very, very long time. And we had a wonderful interaction. More important, discussing immunology was always a pleasure with Dr. Sinus. He has been on the doctoral committee and is for so many of my students, and so he's rather aware, and very good suggestions from him. And students always like to have Dr. Sinus as the chairperson. So thank you, Dr. Sinus. Thank you once again for the kind words. I would also like to thank the Executive Committee of the Indian Immunology Society for inviting me to give this talk during the Golden Jubilee lecture series. A special thanks to the President, Dr. Taruna Madan, and the Secretary, Dr. Balakrishnan, and all the EC members. You have started a wonderful initiative because IIS is something special. It holds a special place in our hearts. We grew up with IIS, whereas our career started, and we were connected to IIS. It was like every year attending the IIS meeting. And those days, there were not many societies. So going to IIS was really one of the main goals during our careers. We met all our friends and colleagues at IIS and wonderful interactions. So it has enriched us academically also. And I hope this tradition of IIS continues. So thank you once again, all of you all. And with these words, I will now, should I start my presentation?
Yes, please.
Thank you. What has happened now?
Sharing screen.
Yeah. Is it, can you see it?
Yeah.
Yes, ma'am.
Okay. So today I'm going to talk about gamma delta T cells and which I call they are the potential game changers for cancer immunotherapy. It's based on the work we have done and also the lot of literature which is coming up on this unique cell type. Of course, this is the place where I've done a lot of work on that track. But before that, just a brief background that I started my career, as Dr. Sainis mentioned, at the Cancer Research Institute from 79 to 83. I then went out to Max Planck Institute for Immunology in Freiburg for the postdoc and then joined back and continued my work both at the Parel campus, and in 2002 we moved to this beautiful location in Kharghar. So that has been a very long journey and a long record of the work with gamma delta T cells from 1987 to 2019. And I don't think it stops here. It still continues in my mind, and I still have a lot of ideas which I love to share with people. So joining Stefan's lab was a wonderful experience. But when I joined his lab, and of course he had come to Cancer Research Institute and he wanted, was looking out for a postdoc. He was a young person then, almost I think around 37 years old, and very dynamic. And T cell clones was a hot topic. And he said that you'll have to work with, you have to make M-Lepre T cell clones. I had no clue, no idea of what it is or what it was and what I was going to face. It was really a very, very challenging project, leading to a lot of frustrations. I had given up my position at CRI. I had just joined as an officer, and those years they were not giving postdoc fellowships. We were not allowed to take leave for more than 1 year. So I had to leave my job. So resigning and going back to a postdoc position and facing such a tough problem was not easy. And so in parallel, I started working on another project that was on B cell growth and differentiation. That is the role of IL-1 in B cell growth and differentiation. So two projects going on simultaneously. One project going ahead fast, the B cell one, which Stefan was not interested, and the mycobacteria project taking sort of a backseat. But I'm not going to tell you the entire history, a lot of struggle, a lot of ups and downs, but finally we got the clones. And we did a lot of work, and these were our first papers to show the role of cytotoxic T cells in M-Lepre. Now that was the year from 84, 85, when Susumu Tonegawa had discovered the gamma delta TCR genes. And during our journal clubs, those days, because we were very close to, the Max Planck Institute was very close to Basel. And so we used to have Polly Matzinger, Charles Janeway coming to us for journal clubs. And journal clubs was held at odd hours at night, after 8:30 or after 9:30 also at times. And everybody was discussing gamma delta, what are this? This is just, something crazy. Only the genes are known, the function is not known, and so on and so forth. And we heard all this debate. But somewhere I felt I got attracted to this cell type. And when I came back to India, I thought I should start working. Yes, and the more the reason was because in TB granulomas, there was histopathological data to show that there were gamma deltas. So that attracted me more towards it. And I knew I'm not going to continue work with mycobacteria. And I was interested in understanding the role of these cells in cancer. Now, the challenge I faced when I came back to work with these unconventional T cells, the gamma deltas, or even we did some work on iNKT. How do I get the probes? Because those days antibodies were not available. There were no probes even to do, there were no flow cytometers. We had one analyzer, Dr. Sainis will know, it was in Dr. Mitra's lab in the TMH building, in the next building of TMH. And it was mostly used for patients. And so very restricted permission to do any work on flow cytometer. And imagine starting work on gamma delta T cells. I wrote, to get probes was very difficult. So I wrote to several scientists if they can gift me the antibodies. It was not easy in those days. It was not like getting the antibodies, getting the antibody itself, posting it, and was a nightmare to get it in the right condition. It was Dieter Kablitz to whom I wrote, and he said, "Oh, Shubha, you're late because I've just given the clone to a company." And I said, "Okay, can you give me a few microliters at least to first show the evidence that there are gamma deltas in oral cancer patients?" So he gave me some small amount, but he said, "Later on, you'll have to buy it from the company." And we started that work with that very small amount of antibody, which was gifted by Dieter Kavlitz. And that was the time when, as Dr. Sainis mentioned, Anupama Lad joined my lab. And I wanted Anu to start working on gamma delta T cells. But I knew that without the probes and without the right infrastructure, it would take her long. So I, with my experience in doing limiting dilution analysis and cloning, I told her, first, let's find out the frequencies of cytotoxic T cells and NK cells in peripheral blood, in tumor, in lymph nodes, metastatic lymph nodes, non-metastatic lymph nodes, and so on. And so I could keep her busy for a year or so by the time I figured out how to get all the antibodies and what was needed for gamma delta T cell work. Everything went on well. Even Anu got some good publications from the cloning work which he did, and we started our projects. So I knew gamma delta to establish myself in the gamma delta field would take long, so I started addressing some other questions, and those were trying to understand the immune dysfunctions in patients. And I've just shown here the basic research projects which we undertook. Several other students, because they were not, it's not only the gamma delta team, but there are others who contributed a lot to understanding of other projects. We got some very interesting translational leads, which are also outlined here. And more important, the experience we gained from all this work. We collaborated with clinicians, and I have done several clinical trials along with the clinicians. So this sort of enriched my experience in doing basic projects and also in taking the leads forward to the clinics. Now, why oral cancer? First, because it is a major malignancy of the country. It is a vexing problem. And the immune scenario, the immune repertoire was not very well understood in oral cancer. Now, if we talk in today's term, these are hot tumors. Hot in the sense they're very well infiltrated with lymphocytes. So when there are so much lymphocytes, the tumor infiltrating lymphocytes, why are they not functional? So definitely they made dysfunctional in the tumor microenvironment. And that's how we decided that we would try to dissect out the reasons possible. Recently, we contributed this review, which is we've almost covered all the aspects of the immune suppressive microenvironment in oral cancer. So as I said, side by side, we were doing both streams, gamma deltas and looking at dysfunctions. So it was Persis who was in my lab, Persis and Dakshini, who did a lot of work on understanding the immune dysfunction by looking at the CD3 zeta chain. This is a main signaling molecule in T cells. And we showed that there is a down regulation of TCR zeta chain in oral cancer patients. We showed the level both at the transcriptional, translational level, and also the contribution of the tumor per se. Is the tumor secreting some factors which are downregulating the signaling molecule CD3 zeta? It took a very long time. Just imagine, Disha, Dakshini, and Persis worked with me till the year 2009 or 10. And we got to know the tumor-derived factor which degrades these zeta when Asif, who joined my lab much later, and I think in 2013, we published this paper that it is the extracellular 2,5 oligoadenylate synthase, OS2, which mediates T cell receptor CD3 zeta chain downregulation via caspase-3 activation in oral cancer. Of course, a lot of help in mass spec we got from Dr. Kanuri Rao, and we were able to then decipher the role of OS2. This was a very interesting lead which we got because zeta has motifs for caspase binding, and OS2 we showed, we chalked out the whole pathway, and we showed how OS2 degrades CD3 zeta. So imagine, so by that time, we had also started work on gamma delta T cells. a lot of work went in because these are very unique cell types. They're only very low in numbers in the peripheral blood. Only 5 to 10% of total T cells are gamma delta cells, and they mediate both adaptive and innate immune-like functions. They were restricted in their TCR repertoire, and the dominant family, the dominant gamma delta chains is the V gamma 9, which pairs with V delta 2, and 90% of the peripheral gamma delta T cells are gamma 9 pairing with delta 2, and a very small fraction, which is V gamma 9 delta 1. Now, the location of delta 1 is mostly in the tissue linings of the body, and where they form the first line of defense. So the mucosal linings harbor delta 1. And what we found that also there is infiltration in the tumors of delta 1. The beauty of gamma delta T cells is they are not MHC, they are MHC independent antigen recognition. So MHC restriction rule is not needed. Upon activation, they produce tons of interferon gamma and TNF alpha. They are highly cytotoxic. They have all the machinery to kill the tumor cells because they show potent anti-tumor effector functions. They're perforating enzymes and so on. They themselves also functions as APCs and therefore play an important role in immune surveillance and in anti-tumor activity. Now, if you look at the cartoon, the ribbon diagram which is shown, the gamma delta T cells variable region very much represents the immunoglobulins. So they're closer to immunoglobulins in the way they capture antigens or bind to antigens. Back in 90s, we didn't know what are the ligands they recognize, how they bind to the ligands, nothing was known. So there was a lot of attempts to understand what are the antigens on the tumors that the gamma deltas recognize. And these were heat shock proteins. They could be heat shock proteins. They could be alkylamines. They were phosphomonoesters, which are intermediates of the mevalonate pathway of cholesterol metabolism, or pharmaceutical agents or drugs like amino bisphosphonates, which indirectly activate gamma delta T cells. Actually, we started working with heat shock proteins much earlier. I think I'll come to that later. Yeah. So as I said, besides the heat shock proteins and alkylamines, it was the bisphosphonates which were important because these are the nitrogen-containing bisphosphonates. And we worked on several of them. We worked on zoledronate, pamidronate, aldronate. But finally, we narrowed down to zoledronate. And this amino bisphosphonates, as I said, it inhibits the farnesyl pyrophosphate synthase in the mevalonate chain. And because of this, there is an upregulation of isopentenyl pyrophosphate upstream. And this isopentenyl pyrophosphate is an antigen which triggers or activates gamma delta T cells. Now, this has opened up several avenues because zoledronate is given to patients to inhibit osteoporosis. It is given to patients who have bone metastasis or skeletal malignancies. And these are readily FDA approved, readily available drugs on the counter. So it's a lot of work or a lot of attention now on zoledronate and gamma delta T cells, beautiful agents if you want to expand gamma delta T cells. And because of the property of gamma delta T cells, they are excellent agents to be used for immunotherapy, both in solid tumors and in leukemias. So now in clinics, you have unmodified gamma delta T cells as well as engineered CAR T gamma delta T cells. And the antigens which they recognize are the IPP, the isopentenyl pyrophosphate, and the HDMAP, the hydroxymethyl pyrophosphate, which is IPP is produced by the mevalonate pathway, while the HDMAP is upregulated by the Romer pathway, which is in bacteria. So these are the two analogs which activate gamma delta T cells. HDMAP being highly potent compared to IPP. Now, over the years, we have asked several questions on gamma delta T cells. What are the ligands? And we have contributed to each of these. Do metabolites of the mevalonate pathway activate anti-tumor cytotoxic functions? What are signaling networks? And we show that there is some crosstalk between Notch and the TCR. Then we were interested in understanding how epigenetic changes regulate effector functions of gamma delta T cells. The tumor microenvironment and the behavior of gamma delta T cells in tumor microenvironment was interesting to us. And the microenvironment was, of course, hypoxic. So this was an area, in fact, one of my last students worked on how understanding the role of hypoxia in dictating the effector functions of gamma delta T cells. And finally, we are interested in understanding the crosstalk of mesenchymal stem cells and gamma delta T cells in tumor microenvironments. The last part is still, I'm not be talking about it today because we still have to publish that part of the work. So coming to the story, when we started, way back, we published an International Journal of Cancer in 1999. Anupama and Louis joined later, and we showed that heat shocks proteins are constitutively expressed on tumor cells. And we focused our attention on HSP60 and HSP70. And it was very, very difficult those days to isolate gamma delta T cells. You know, we didn't have, of course, very quickly we got the immunomagnetic sorting done because those were just come out in the market. But before that, you know, the culturing was to be done with Dowdy cells. And if you incubate peripheral blood mononuclear cells with Dowdy, you get again outgrowth of gamma delta. Much later did we realize that Dowdy cells express tons of heat shock proteins. And that's how we started working more on heat shock proteins. And we showed that these activated gamma delta T cells are able to lyse, and this lysis is mediated by V gamma 9, V delta 2, because this is percent inhibition of cytotoxicity. The cytotoxicity gets inhibited if you block V gamma 9 delta 2. And if the tumors are treated with HSP 60 or 70, there is the cytotoxicity decreases, indicating that the heat shock proteins are recognized by V gamma 9, V delta 2. Heat shock proteins expressed on tumor cells. We addressed this question in oral cancer. We also started looking at esophageal cancer. And in esophageal cancer, Louis Thomas, who was in my group, there are a lot of studies on understanding how gamma delta T cells migrate to the tumors. So looking at a lot of adhesion molecules and looking at their interactions and their usage by gamma delta T cells. In those days, he was able to separate the gamma 9 delta 2 and V delta 1s, and then looking at these subsets independently in their characters and the expression of adhesion molecules. At the same time, there's a lot of discussion now that gamma delta T cells, which are stimulated, undergo activation-induced cell death. And it was Nilangi Atre who joined my group, and I gave her this project to understand what causes apoptosis of gamma delta T cells. And very interesting finding, again, we published in International Journal of Cancer in 2006, that it is gamma deltas do not use the FAS-L pathway for apoptosis, but they use the nitric oxide released by the activated gamma delta T cells via activation of caspases and mitochondrial damage. So the nitric oxide and the NOS, nitric oxide synthase and NDNO were involved in bringing out apoptosis of gamma delta T cells. So the story continued then. And again, I'm showing you the same cartoon just to emphasize that once you give amino bisphosphonate to the tumors, there is an upregulation of IPP. And this IPP is the target antigen for gamma deltas. As I said, this is the IPP is generated through the mevalonate pathway. It is also generated through the Romer pathway or DOXP pathway. The HMBP, which is generated, is 1,000 times more potent in activating gamma deltas. So you can understand in the bacterial infections, and they have shown that the gram positives and the gram negatives, the differences in the expression of HDMAP and the activation of gamma delta T cells. So we started profiling. This was not all done at the same time, but to look at whether gamma delta T cells are present in the peripheral blood of cancer patients, are they present in the tumor infiltrates? And these were taking surgically excised tumors and looking at the infiltration. And we could show that in oral cancer patients, there were high levels of gamma deltas in the peripheral blood, and which were still further increased in the tumor infiltrating lymphocytes. All this work done by flow cytometry using V delta 2 as the probe. And in pancreatic cancers, of course, now the numbers have increased, but again, the same picture. But it is not the same everywhere. It doesn't mean that gamma delta T cells are there in the peripheral blood of all the cancer patients or they're present in the tumors. Look at the case of gallbladder cancer. And in gallbladder cancer, the levels are not very different in the healthy individual, peripheral blood, and the tumor infiltrating lymphocytes. In colorectal cancer patients, in fact, we got much less infiltration. So it differs in different malignancies. And as I said, the hot and the cold tumor concept makes it very interesting to understand what stimulates or activates gamma delta T cells in some and not in others. During that time, Swati Dhar joined my lab, and we decided to treat the tumor cells with amino bisphosphonates and see whether they become susceptible actually to the gamma delta T cell kill. Now, you can refer to this paper. It's, in 2010, it was published in Cancer Immunity. There's a video attached to this tumor. For those who can watch the video. And it was interesting those days for us to show that how gamma delta T cell actually killed the tumor targets once the tumors were treated with zoledronate. It's a huge difference. You take untreated tumor cells and you take treated tumor cells. The treated tumor cells are aggressively lysed by gamma delta T cells. So this remained like, as a gold standard in my lab to use zoledronate treated tumor cells when we look at addressing the cytotoxic effector functions of gamma delta T cells. Again, we went in deep to understand what are the molecules, and so we inhibited gamma delta T cells express NKG2D, and through which they bind to MIC-A, MIC-B, which is expressed on the oral tumors. And so we use these specific antibodies to inhibit the subset, the gamma 9, when you inhibited the subset, you got an inhibition, and when you inhibited along with NKG2D, you got an inhibition. So this directly showed that it is the NKG2D which binds to MIC-A, MIC-B on the tumors. I'm not describing all the experiments which bring about lysis of gamma delta T cells. Now I want to tell you today, we know much more, and it's from Cablets has actually presented a review. There are several other groups who have contributed to the understanding of how gamma delta T cells recognize the phosphoantigens. And it's because of the molecules which are called as butyrophilins. These butyrophilins are present on the epithelial cells and on the tumor cells. And there is an intracellular domain which is called the B30.2 domain, which actually binds to the phosphoantigens and then brings about a conformational change in the other molecules, the BTN2A1 and 3A1, because of which they get hooked on to the V gamma 9 chain and activate the gamma delta T cells. So now this molecule is known, the endogenous 30.2, where the phosphoantigen binds, and then the BTN bringing out the change in the BTN2A, 3A1, which then activate the V gamma 9, V delta 2 TCR to activate signal transduction. And now there are a number of papers which show that tumors express these butyrophilin molecules. So we had fairly established ourselves in the gamma delta field when one day I get a call from Netherlands, from Unilever, and this was a group of scientists from Unilever who were interested in understanding if black tea and black tea components can activate gamma delta T cells. So we had several meetings, and they said that since you have the model with you, you have purified gamma delta T cells, you know how to isolate. Can we look at this? And we did a collaborative project with Unilever, and we showed that the black, and it was not only the black tea components, they were also interested. I don't know if you all remember, but there was an advertisement from Unilever that immunity makes a difference, and where they showed an old lady and a young person drinking tea, and with a lot of other herbal components added to it, ashwagandha, shatavari, and all, so many. So we tested about 12 to 13 of those compounds along with black tea, and showed that the black tea components have the ability to stimulate gamma delta T cells. And it was very interesting to note that this stimulation occurs very similar to how bisphosphonates behave. So Nirmala Shanoj in my lab did all this work, and we showed that black tea fractions can, if in PBMNC you throw black tea fractions, they can activate gamma delta T cells, they can mediate potent toxicity. We looked not only at ethylamines, we looked at all the other alkylamines, sec-butylamine, isobutylamines, and there was sort of hierarchy in which they activated gamma delta T cells. So this was another interesting aspect, and moreover, sort of a nutraceutical suggestion to have it as a nutraceutical to activate gamma delta T cells. Then the most important feature we thought is, okay, gamma delta T cells are highly cytotoxic, they kill tumor cells. Are they able to mediate ADCCs, that is antibody-dependent cellular cytotoxicity? And for this, naturally, you need an antibody. And as Dr. Sainz mentioned, I worked with Dr. Gangal, and as a team, we were able to make antibodies. I made antibodies to alpha-fetoprotein along with Dr. Kishore Amin, who was in a group. We also made antibodies to oral squamous epithelial cells. And these antibodies, we are doing a lot of immunohistochemistry to look at different tumors from buccal mucosa, tongue, and all other sites. But with my background of gamma deltas, I was interested in understanding what is the antigen they recognize. And before that, we started doing imaging studies. So this is a SPECT imaging where we transplanted a tumor into nude mouse and showed that we could image with this monoclonal antibody. We started doing the ADCC experiments, taking purified sorted gamma delta T cells using tumors, taking both fresh tumors as well as cell lines, and again looking at the same logic, treating the cell lines with zoledronate, and these are untreated targets, untreated cell lines. Incubate the gamma deltas, the target with the monoclonal antibodies, and throw in gamma delta T cells. And this is what you see. This is the target alone with gamma deltas. This is the level of kill. You add the monoclonal antibody, you get a huge increase in cytotoxicity. And this was the control mouse immunoglobulin. This was a dose-dependent response which we got. Now look at the right side of the figure. These were zoledronate-treated tumors. So you compare the red to the red across and the blue to the blue, and you see a massive increase in cytotoxicity once the tumors get coated with zoledronate. So zoledronate-treated tumors having tons of IPP, getting beautifully lysed by the ADCC, which is mediated by gamma delta T cells. And of course, this is in the surgically excised fresh tumors. So this was a wonderful property of gamma delta T cells to mediate antibody-dependent cellular cytotoxicity. To move further, we were interested in understanding actually what is zoledronate doing to the tumor cells. What are the proteins or other differentially expressed proteins in zoledronate-treated tumor cells? And I think Dr. Sainz will be more familiar because this is Swati Farke's work from my lab, who was doing, she did a lot of proteomics, and she used ITRAC labeling and LCMS to identify the proteins that were expressed. Now, I'm not going through the whole data because there are a lot of tables in this, but I've just summarized this in this cartoon, which shows that once you treat tumors with zoledronate, there's downregulation of lipid metabolism, there's EGFR downregulation, there's cell cycle arrest, there's cytoskeletal disorganizations, then downregulating of the DNA repair system, apoptosis, there's copper toxicity because the cut C and the cut A, the ones which throw off and throw in, so a lot of copper gets accumulated, downregulating of vesicular traffic and protein degradation, and most importantly, upregulating the IPP synthesis catalyzing enzyme, phosphomevalonate carboxylase, and IPP So this actually fitted well in place with our logic that it is the IPP that accumulates and could be a target for gamma delta T cells. Now we validated this, and Swati did some very nice experiments, and she showed these are the untreated. We looked at the expression of actin because the cytoskeletal disorganization. We thought we'll look at actin, we'll look at keratin, tubulin, and the actin filament, that organization is lost, and moreover, the molecules involved in actin polymerizations are downregulated. So the ARP2/3 complex is all downregulated. So you can clearly see the filaments here, and they are all disturbed in ZOLT treated. In keratin, and specifically we looked, because these are epithelial tumors, we looked at keratin 8, and the network of keratins was sort of thinning of the network of keratins. So that were definitely keratins also were affected. Tubulin, we did not see much changes in tubulin, though the proteins levels increase. There was not much change in their organization as far as, or we could pick up with confocal. So we could validate definitely that there was change in the cytoskeletal structure when the cells were treated with zoledronate. We also then looked at the cell cycle, and it was, and all these proteins involved in cell cycle were affected, and we saw that zoledronate arrested breast cancer cells clearly in the S phase. So we could validate our proteomics data. A whole lot of work has gone in because it's Swati's thesis, which looks at all these changes. We thought, why not address the question in patients? So again, those days we were discussing, and we discussed with Dr. Sudeep Gupta, with Dr. Vani Parmar, what would be the best patients, and they suggested that we look at women, postmenopausal women, who have bone metastasis, who do not have bone metastasis, take these two cohorts, and see if there is an increase in gamma 9 delta 2 phenotype. So we extensively profiled these women, their lymphocytes for the expression of gamma deltas, looking at activation markers, CD69, RANKL, CD25, and these are untreated. These are women treated with zoledronate, who were given zoledronate, and you see a massive increase in the activation status, gamma deltas expressing CD25. And what was interesting is there was an increase in the effector memory T cells, the TEMRA phenotype was increased in these women. Now, and around us, there were also reports coming up from patients with multiple myeloma who are given zoledronate treatment to inhibit the skeletal metastasis. So more and more proof was emerging that in vivo, zoledronate expands the gamma delta T cells, which have cytotoxic potential. So zoledronate has a dual role. It has a direct action on the tumor cells, which I showed you all. It disturbs the cytoskeletal structure, DNA repair, and several other things. And it also has a direct effect on gamma delta T cells. And so zoledronate would be a wonderful pharmaceutical agent to be given to expand the gamma delta T cells, and which is being now used in immunotherapy, even along with CAR T cells. Further, we thought we should do some preclinical animal work. Those days we didn't have SCID facility. We had nude mice, and we used nude mice, transplanted them in tumors. But the difficulty we faced was to get gamma delta T cells every time for every injection from the same donor. It was not easy. You know, you need the amount of gamma deltas you need to isolate as freshly isolated gamma delta T cells. You need at least 100 ml of blood every time. And it was a daunting task to get this amount. So we said we'll use another, we thought, let's have a proof of concept. We give gamma deltas in vivo and then monitor doing histopathology to look at what happens to the tumor cells. So we took this, and we were able, these are short-term experiments. We monitored them for 48 hours, 72 hours. We could do these experiments only for a week. And what we showed by tunnel assay, this is a positive control. And just look how beautifully you can see the dying tumor cells once gamma deltas were injected in the presence of zoledronate and IL-2. Then the story continued, and I met Dr. Rinti Banerjee from IIT, and she was also interested in something in immunology, and I told Rinty that with your expertise, you can make nanoparticles of bisphosphonates. She was very much interested, and it was around this time. So like we had been discussing this for almost a year, but then came a call from DBT for UAE, the Unit of Excellence, and we formulated a group in which we had Dr. Rinty Banerjee, myself, and Sanjeev Galande. And so we said we'll address a common problem of looking at gamma delta T cells. And Rinty started making her student making the CZ nanoparticles, and we were characterizing these nanoparticles. And we could see beautiful outgrowth. If you take PBMNCs and throw in these nanoparticles, you can get an outgrowth of gamma delta T cells. These gamma delta T cells, which were activated with the nanoparticles, actually could augment cytotoxicity, again in a dose-dependent fashion. Though we didn't see, these were Xolairide treated and these were the CZ nanoparticles treated. We didn't see a huge difference between the two. So then Rinty wanted to modify, and they wanted to still work and make, but unfortunately we couldn't continue as Rinty passed away, and this project sort of halted. We were able to do some work, but it's very unfortunate that we couldn't take this study further, which would have been a wonderful study. Rinty's student worked in my lab, and we did some experiments on imaging, and which showed that there was a reduction in the tumor, or reduction in the tumor volume. But there were about 4 or 5 types of nanoparticles which they had made, but we were not able to take the study ahead. We looked at alternate methods, and we thought we'll do in vitro imaging. And I was more keen on understanding if we can measure intracellular levels of isopentenyl pyrophosphate. That would be a more quantitative assessment of the gamma delta function. So this in vivo imaging, and this was Trupti Pradhan, she's a scientific assistant working with me, did this experiments, coating the tumor cells, putting in gamma delta T cells and imaging them. And clearly these are untreated and these are the Zol treated, where you start seeing clearance or killing of the tumor cells using all these blocking agents. So this was fine, but my main aim was to have an assay, and which Trupti worked. She used the LCMS, and we standardized the protocol for measuring the metabolite IPP. And then we showed that treatment with Xolairide upregulates treatment with those. We used those nanoparticles, and we did this when Rinty was not there, but the nanoparticles were with us. So we did some few experiments to show that we could see an augmentation of IPP. You give mevastatin, the upstream inhibitor, and the levels drop. You treated with Xolairide in the presence of mevastatin, the levels are low, and treated with Xolairide nanoparticles. So we have this method in hand where we can measure intracellular IPP. I thought this would be a beautiful handle when even patients, because bisphosphonates are given, there are toxicities which are also associated. Jaw necrosis is one of them, very common. But breast cancer, these are given for osteoporosis, it's a long-term treatment. For cancer patients, not such a long treatment. But it would be nice to have a method where we can, intracellular levels of IPP can be monitored and efficacy of Xolairide can be established. We then wanted to look, since Xolairide also has an action on bones and on the osteoclast. So we wanted to study whether the osteoclastogenesis is some way affected by Xolairide, and what happens is there's a crosstalk between bone and gamma delta T lymphocytes. So this axis of gamma delta T cells and tumors, a lot of work went from the lab. This was something new. And it is again Swati Phalke. Actually, her project work coupled these two things, looking at what Zol does to the tumors and looking at the crosstalk of osteoclasts and gamma delta T cells. Again, nobody in the lab had worked on osteoclasts. We had no idea of how to start. So I was looking and I realized that Dr. Mohan Wani from NCCS was working with osteoclasts. I think he had published by then. but his was a murine system. So but I sent Swati to his lab to see how osteoclasts, to establish the osteoclast cultures. And she came back, she did beautiful work on generating osteoclasts. But the task was to make human osteoclasts. And this is not very easy. And using a cocktail of MCSF and Rankel, she established beautiful multinucleated osteoclasts. She used the right markers of vitronectin receptors to characterize these osteoclasts and the F-actin ring formation, which is seen in these osteoclasts. So we were very confident that we can go ahead because since the osteoclasts were ready. The next step was to standardize the bone resorption assays. And for that, we searched and we found that using OAS plates, basically this is a calcium phosphate matrix which resembles the bone. You can look at bone resorption on these, do the assays for study bone resorption. And Swati standardized very nicely this work. And we showed, we published that activation status of gamma delta T cells dictates their effect on osteoclast generation and bone resorption. What do you mean by that? We took two sets of gamma deltas. One, gamma deltas that were freshly isolated directly from the patients. Now, this is not an easy task. You need a lot of blood to get enough amounts of gamma deltas with which you can start or establish such co-cultures. And then the activated, which is taking out the gamma deltas, throwing in bisphosphonates or BRHPP, which are analogs of IPP, and then culturing them for 12 days in culture and then looking at the effect on osteoclasts. We decided to use both. And to our surprise, what we found was there's a big difference when you use freshly isolated versus activated gamma delta T cells. And the entire dynamics of bone resorption changes when you use freshly isolated and the activated gamma delta T cells. There is a lot of osteoclastogenesis seen, promotion of osteoclastogenesis if you use the freshly isolated gamma delta T cells, because when you looked at the cytokine dynamics, again, this was a very complicated experiments with several groups, but to cut the story short, freshly isolated gamma delta T cells secrete IL-6, while the activated gamma delta T cells secrete more interferon gamma. So more interferon gamma are the ones which inhibit osteoclastogenesis, and those which secrete IL-6 are pro-osteoclastogenic. So this unraveled a new phase or new picture of gamma delta T cells and the way they behave when they would come in contact with bone cells or bone remodeling. Now, what was very interesting is just last week, I got hold of this paper, or with this, it's hot from the oven, I would say, that a paper has come in Science Advances, which says that gamma delta enriched CAR T cell therapy for bone metastatic castrate-resistant prostate cancer. And this study has quoted our reference because, as I said, that was the only work in the field of gamma deltas which showed how osteoclastogenesis can be promoted or inhibited using gamma delta T cells. And reference 55 years our paper in this article. And I'm sure this will be, this would encourage the CAR T field to take ahead gamma delta T cells as therapeutic agents. Sorry, Dr. Sinus, should I continue? How long? No, I can't. Taruna, how long?
Yes, yes, ma'am. Please continue. I think sir is on mute. Sir is not realizing that. Yeah, please continue, ma'am. It's a very interesting work.
Yeah.
We would all like to.
Yes.
Okay.
Yes, Dr. Chibrankar, you can continue. We have
okay,
time.
Thank you. So now we showed the different functions of gamma delta T cells. We are now interested at what regulates the function of gamma delta T cells. And that was the time when Dimpu Gogoi joined my lab for a PhD. And I was reading about the literature, and there was no reference of the effector function, how Notch regulates effector functions. But there are a lot of papers coming on Notch regulating the lineage discrimination between alpha, beta, and gamma delta. Again, these papers were on mice, and you cannot correlate the mouse work with humans. There's a lot of difference. A difference to that extent that mouse gamma deltas don't recognize phosphoantigens. So the mouse literature, though I was reading it, I knew it is not. It may be very different scenario in the humans. In the humans, I told Dimpu that, Dimpu, your PhD work is going to be on Notch. And so he went and I think he scanned the literature. He came back and he said, Madam, but nobody has worked on this. I said, why would you be here then? You know, if everybody has worked on it, I wouldn't give you that as a project. So I can imagine his feelings of a new PhD student with no reference to fall back on, and his mentor giving him a project which is so challenging. But he took equal interest, and we started first profiling the gamma delta T, trying to see whether they express notch ligands and notch receptors. And we used both the, we looked at the RNA and we looked at the protein expression. And as you all know, very briefly, when notch binds to its receptor, there's activation of ADAM proteases, which cleaves the extracellular membrane, and the intracellular gamma secretase gets activated, which releases the notch intracellular domain, which translocates to the nucleus and initiates both canonical and non-canonical notch signaling. So this is in brief. If you use a biochemical inhibitor like gamma secretase, gamma secretase inhibitor, you can prevent formation of NICD and translocation to the nucleus. So we made use of this strategy in our experiments, and we showed that notch. The interesting thing was notch is expressed on gamma delta T cells, and when we give IL-2 plus BRHPP, which is an analog of IPP, you do see an increase in the notch. And MYC is known to regulate NICD. So we also looked at CMYC to regulate notch. So we also looked at expression of CMYC, which is enhanced in the presence of BRHPP IL-2. And you give gamma secretase inhibitor, and you start seeing decrease in the levels of notch intracellular domains. We went further and we tried to look at what if you block the using gamma secretase, and we used also the siRNA approach to block the notch receptor, and we clearly showed that targets treated with zoledronate are inhibited when you get notch siRNA, and this reflects in the loss of the notch intracellular domain. So this fitted very well that notch plays an important role in the cytotoxicity mediated by gamma delta. It regulates the cytotoxicity of gamma delta T cells. It regulates also the antigen-specific proliferation of gamma delta T cells. We looked at the expression of CD107, degranulation, perforin, granzymes, and notch regulates all these functions in gamma delta T cells. Yeah, here it is. I thought it's not there. So we looked at these, the profile of the various transcription factors, TBET, EOMs, and we see that in the presence of the gamma secretase inhibitor, this is downregulated. So this was the paper, and when we communicated this to Journal of Immunology, where it got accepted, and sort of emphasize some work which we were doing in alpha beta T cells, and which Asif in my lab was looking at, and he showed that notch signaling has the ability to alter the proximal and distant signaling events in CD3 alpha beta T cells. But the way they work in the two is different in alpha beta and the gamma delta T cells. I'll talk about it a little later. This again, I come to the project now, which the DBT UOE project, where Rinti was there and Sanjeev Galande also was my collaborator. And we decided that we'll do a high throughput transcriptomics to look at what happens to the gamma delta T cells when they're stimulated with different antigens. And what are there any differences in the signaling pathways? So in this cartoon, you can see we took gamma delta T cells alone. And mind you, these are all freshly isolated gamma delta T cells. So to get them in huge numbers for the experiments is not easy. Gamma delta plus IL-2, gamma delta plus HD map, which I told you is intermediate, the Romer pathway, gamma delta stimulated with IPP, gamma delta stimulated anti-CD3, which is the classical trigger for the T cells, and then using gamma secretase inhibitor to block. And the final picture we showed was that the distinct and overlapping transcriptional programs in gamma delta T cells after stimulation with HDMAP, IPP, and CD3. Now, IPP, the pathways affected are cell cycle and interferon gamma pathways, more abundant in IPP treated cells, while cytokine production and lymphocyte migration gets enriched in HDMAP. I told you the HD map is 1000 times more potent in activating gamma delta T cells compared to IPP. And the interesting part was, you stimulate gamma delta T cells with anti-CD3, they secreted high amounts of IL-17A compared to HD map and IPP. In fact, HD map and IPP, the IL-17 secretion was low, but with anti-CD3, it was high. And this actually is a boon to those who are doing in vitro activation of gamma delta T cells for therapy, because you are not going to then throw gamma deltas in a Th17 differentiation pathway, but you're going, when you stimulate them with the respective antigens. And this study brought out another important point, that Notch blockade with GSI down-regulates interferon gamma in GRAN enzyme, confirming the crosstalk between TCR and Notch signaling in gamma delta T cells. After having done this, we were interested more also in looking at epigenetic regulation of gamma delta T cells. And here again, this was a new story for us, because, and Sajad, who joined my lab, took up this responsibility, and we addressed the role of epigenetic regulation of gamma delta T cells. And the focus was to understand the question in the clinics, where HDAC inhibitors are given to patients. They were in clinical trials. They're facing a lot of issues with toxicity. And moreover, all the work which has been done or reported mostly is for HDAC inhibitors is on their effect on the tumor cells. How and look at pathways in tumor cells which are affected by HDAC inhibitors. There were a few papers which came up for CD8 and NK cells, and we thought it would be interesting to do a very comprehensive analysis of their role on how these HDAC inhibitors affect the gamma delta T cell functions. And this is a list of all HDAC inhibitors which are in various clinical trials. And what we did, we selected the class 1 and class 2 HDAC inhibitors, which are pan inhibitors, the valproic acid, the TSA, and the SAHA. These three, this is the vorinostat, the valproic acid, and the SAHA, I forget the full form. It is suberynyl anilide hydroxamic acid. So we took these three, and we started looking at how they affect antigen-specific proliferation of gamma delta T cells using HD map as the antigen. And with all the three, you see clearly that the antigen-specific proliferation diminishes in the presence of the when you add the HDAC inhibitor. Now, if this is happening, if a specific proliferation, what is happening to the levels of P53 and P51, P21, which is downstream target of P53, and they definitely increase, which is expected. So downregulation of antigen-specific proliferation really was something surprising, and we decided to pursue this further. And we looked at all the cytotoxic functions against a battery of cell lines. We looked at expression. This is a very big paper, extensive work done in Frontiers and published in Frontiers in Immunology, and those who are interested can have a look at it. We looked at TBET, EOMs, and looked at all other functions of gamma delta T cells in the presence of these HDAC inhibitors. And we also did chip assays to show that HDAC inhibitors decrease the acetylation on promoters of perforin and granzyme B. So why was this happening? Why were they not responding, or why was proliferation cytotoxic functions being affected? We decided to monitor looking at the checkpoint molecules, the PD-1 and the PDL1. And to our surprise, we found that there's a dose-dependent increase in the expression of PD-1 and PDL1 in the presence of the HDAC inhibitors. This I've shown for VPA, but it's also true for all others. So somehow the cells were getting into a state of exhaustion because of very high levels of PD-1, PDL1. The next we thought was, if we use a combination of HDAC inhibitors and blockers for PD-1, can we rescue this function? And this is the cartoon which shows, this is the cytotoxicity which you get in the presence when you add, this is the third one. So you have the gamma delta IL-2 HD map and anti-PD-1. Anti-PD-1 alone does nothing. You add to it the HDAC inhibitor, the cytotoxicity goes down, and then when you give both the VPA along with anti-PD-1, you can rescue the function. So everywhere the function gets rescued if you use a combination of anti-PD-1 along, or you use anti-PD-1 along with the HDAC inhibitor. So this was very interesting for us, and also now in the protocols which are coming up in the clinics, they're using combination therapies of HDAC. There's a phase 1 trials now initiated using checkpoint inhibitor antibodies and HDAC inhibitors. Now to the immunologist, you all know the 3 E's of immunoediting, and as the immune system has an anti-tumor role, it may also have a pro-tumor role. It can promote tumorigenesis. Again, this was an interesting question in our mind, and we wanted to know what is the role of gamma delta T cells or the subset of gamma delta T cells which could be pro-tumorigenic. 2 people contributed to this work. One is Rishikesh Patil. I think he is today online, and wonderful work done by Rishikesh. And nice to say that actually now he's involved in the ImmunoAct, and he's working along with Rahul Purwar in his company. And the second was Shalini, who did the work on hypoxia and how hypoxia drives a pro-tumorigenic phenotype of gamma delta T cells. So first I'll tell about Shalini's work because she addressed the role of hypoxia. Again, a lot of work went from Shalini because the entire thing had to be standardized using the hypoxia chamber, exposing cells to normoxia, hypoxia, looking at the phenotypes. And in short, I can tell you, all this is published. Expansion of gamma delta T cells upon TCR stimulations remains unaltered. So we were expecting, that hypoxia may inhibit gamma delta T cell proliferation, but doesn't happen. The proliferation of gamma delta T cells, the way to look at antigens is just fine. It does not alter the activation status. So we looked at CD69, CD25, no change in them after hypoxia. Then we decided, this is again looking at antigen-specific proliferation with HDMAP, anti-CD3 and others. No, antigen-specific proliferation also is not altered. What is it that is altered? It profoundly disturbs the cytotoxic potential of gamma delta T cells. Now, these experiments which we did were using Zoll-treated targets, using co-culturing Zoll-treated targets with gamma delta T cells, and then using normoxia and hypoxia. So 1% versus 20% oxygen. Then looking and comparing the results. So hypoxia exposed gamma delta T cells exhibited reduced tumor-directed cytotoxicity, which is clearly seen. Just a quick glance shows in normoxia, good cytotoxicity, in hypoxia, it is reduced. We did this across tumor targets which were Zoll-treated. Again, it is the same. Those in normoxia did well. Those with hypoxia, there's a decrease in cytotoxicity. This is using both gamma deltas from healthy donors, gamma deltas from oral cancer patients. All the story goes the same. You expose them to hypoxia and the cytotoxicity is decreased. Then the most difficult part in which we thought, and we designed an experiment, and I'm not showing you all the data because it's difficult to accommodate all that. Just telling you the logic. We took gamma deltas as effectors under normoxic conditions and saw their ability to kill normoxic targets, tumor targets. On one hand, we took hypoxia exposed gamma delta T cells, and which is standard, and hypoxia exposed targets. And then we did crisscross experiments, taking normoxic gamma delta T cells and putting them with hypoxic targets, and taking hypoxic gamma delta T cells as effectors and incubating them with the normoxic targets. Now, this whole assay, culture conditions, the 4R assay conditions also we did under normoxia and under hypoxia. So different combinations. And the underlying story is the same, that hypoxia exposed gamma delta T cells exhibit reduced tumor-directed cytotoxicity. So what is it? What are the changes? These are really very not easy experiments to do. And Shalini did with both fresh tumors and cell lines, and she showed that there is a reduction in the calcium efflux in hypoxia-exposed gamma delta T cells. It will be difficult to read this graph. You can follow the dotted lines and the straight lines which are there, but it clearly shows that the hypoxia-exposed gamma delta T cells exhibit reduced calcium efflux, and also the degranulation, that is CD107 expression, is decreased in them. Then we decided what is happening, and because in our mind it was that if we do a gene expression profiling, what are we going to see with gamma delta T cells which are under hypoxia and which are exposed to under normoxia and activated with different antigens, and along with CD3 as the standard way of stimulating them. And here you can see a signature which clearly evolved is the Th17. These are gamma delta T cells under normoxia, and these are the gamma delta T cells, both normoxia and hypoxia. Yeah. So this was the signature that Th17 differentiation was a striking feature. Then we looked whether these cells which are exposed to hypoxia and normoxia express the delta 17 gamma delta 17 phenotype, and they do. There are high levels. They secrete high levels of Th17, and which is also seen with ROR gamma T, the transcription factor which is present in gamma delta T cells. So this clearly established that hypoxia drives a Th17 phenotype in gamma delta T cells, and again it also showed that IL-17, that anti-CD3 stimulated IL-17 expression in gamma delta T cells significantly increased upon anti-CD3 stimulation, but not with HDMAP. So CD3 and the standard antigen, the phosphoantigens, the way they activate the receptor is different, and you can see this different. Of course, now there are very interesting papers coming out which talks about the receptor stabilization, because there is the anti-CD3 needs NCK to be recruited to the plasma membrane, and which stabilizes CD3 conformation, while this is not needed in case of phosphoantigens. So the other side of the story, and which we called, we were invited to write this article, and we wrote about the Jekyll and Hyde story of IL-17 producing gamma delta T cells, and a very beautiful work from Rishikesh on IL-17 producing gamma delta T cells induce angiogenesis and are associated with poor survival in gallbladder cancer patients. We were looking for a model. Where do we study this? Where do we study the IL-17 phenotype? So we're looking for tumors, and we thought it would be interesting to look at a tumor which grows on the background of inflammation, and gallbladder cancers are the ones. They have a background of gallstones or cholelithiasis, where the microenvironment is inflamed. And so we submitted this as a project to DBT for a center of excellence, and it's a collaborative project between Dr. Dixit, who's now the director of the CCE at ATRAC, and our lab. And this is Dr. Dixit's work, where he was doing the common genetic variation risk of gallbladder cancer in India. Genome-wide association studies comes up with the ABC transporter, which plays an important role. What we were doing, we were looking at the immune profile. And Rishikesh did this work, and he showed that the delta 17 phenotype is high in the tumor infiltrating lymphocytes of gamma delta T cells. He later showed that these delta 17 this delta 17 cells secrete, coming in between, delta 17 cells secrete VEGF, endothelial growth factor, which promotes angiogenesis. And then we decided to do a complete angiogenesis array, and to look at both the angiogenesis inducers. And so this is the angiogenesis, protein angiogenesis array, where you took the gallbladder cancer cells. And the most challenging task was to isolate the delta 17 gamma delta T cell. You can imagine we were struck. It's not easy to isolate gamma delta T cells. To isolate delta 17 was even more challenging. And I must say, Rishikesh did this very nicely, standardized the protocol of isolating delta 17, checking their purities, and showing that they produce a lot of pro-angiogenic factors, which are inhibited if you give anti-IL-17. He did the CAM assay, and he also did the array profiling of these cells. since we had this cohort of gallbladder cancer patients and the survival is very short, not very long in these patients, we follow up these patients for 50 months and we looked at the overall survival looking at specific phenotypes, the delta 17, and we showed, of course, then we had to use an arbitrary cutoff where we showed that those who were expressed 2 times more were said to be high producers of delta 17 and low. So we showed that those with low expression of T deltas, delta 17 survived longer compared to the high. If you have low interferon gamma, the survival is low. So we did with delta 17 producers, IFN gamma producers, and overall survival with total IL-17 and interferon gamma. So this clearly told us that delta 17 are protumorigenic and they are predictors of poor survival in patients. I'm not going to talk on this because later Asif, Rishikesh together showed they dissected out the entire network, immunosuppressive network, which is mediated by myeloid-derived suppressor cells. And again, this is a whole story on myeloid-derived suppressor cells, characterizing the myeloid-derived suppressor cells, monocytic and granulocytic, the PMN myeloid-derived suppressor cells and their functions, and showing that they promote Th17 differentiation oral squamous cell carcinoma. Now, all this story, what I told you, was on solid tumors. We thought it is time we address, actually, the work started when we were doing oral tumors. And Jyoti Kode, who works in my lab, who was my student and later my colleague, she was involved in this work. And so was a PhD student, Dr. Gauri, who did a lot of work on, who continued the work on this profiling of looking at TCR gene rearrangements. Now, when we started addressing the question in leukemias, we thought it would be interesting to address the question in T acute lymphoblastic leukemias because they are clonal malignancies. So they're clonal, alpha beta clonal or gamma delta clonal. They can be a mixture. It's not that they're segregated as alpha beta and gamma delta. So we started doing profiling and we started looking at, and that's very interesting. I must tell you the story. We were working with our clinicians who were, Dr. Banauli was majorly involved, and they were treating patients on an MCP 841 protocol, which is a standard protocol to be given to these patients. And frankly, they were not very interested when I told them that let's profile for gamma delta and clonal gamma delta and alpha beta, because see, they are interested in how is this going to make a difference to our patients. But we continued and we showed that through a series of papers, we showed that 30% of the Indian pediatric TALL patients are clonal gamma deltas. We had meetings and they said, oh, very nice, very interesting work. But what do you do with this? Okay, 30%, they are all put on MCP 841. We are not profiling them earlier to see. So I said, okay, we'll not give up. We followed them up. We followed these patients up for 100 months and we showed that those with clonal TCR gamma delta survive longer and those which are clonal alpha betas die early. So the question now is such an aggressive protocol really required for patients who have clonal gamma deltas in their blasts? And I think somebody should seriously take up this question because there is such a clear picture imaging. And then we did gene expression profiling to see if TCR alpha beta clonal are different from TCR gamma delta clonal. And yes, with respect to leukemia associated genes, respect to apoptosis related genes, and several others, they stand out as a distinct phenotype, genotype in the TALL or T acute lymphoblastic leukemias. Gauri and Jyoti took the story further, and this is all Gauri's PhD work. And then she showed that along with TCR, if you use CDKN2A, CDKN2B, because she was looking at also the copy number variations in these genes. And since we had this cohort of clonal gamma deltas, we had the samples with us. She continued this work and showed that this combination would be an interesting prognostic biomarker for TALL patients. And something which I don't know if Gauri is in the audience, but if she's listening, this paper, she has done a lot of work on looking at the immune synapse because we were interested if gamma delta clonal TCRs are giving them a survival advantage there must be something different in their functions, in the way these blasts interact. So we started doing immune synapse. She has contributed a lot of work, and we will be communicating this paper soon. This story has been taken ahead by Gauri, and also she has looked into CMV reactivations in patients in bone marrow transplant patients, and where she has shown that V delta 1 phenotype is important. I'm not presenting that because that's not published work, but a whole lot of work done also in bone marrow transplant recipients. So to summarize, what I have spoken or given a bird's eye view is gamma delta T cells exhibit potent cytotoxicity, mediate ADCC against solid tumors, against leukemias, blasts, and therefore I feel they're ideal candidates for combination immunotherapy using amino bisphosphonates. There is a pro-tumorigenic phenotype, very well characterized, the T delta 17 phenotype. And we also showed that PD-1 blockade improves the anti-tumor effector function of HDAC inhibitor-treated gamma delta T cells, which results in increased expression of granzyme and LAMP1. And the future protocols, chemotherapeutic protocols for gamma delta T cells should make use of these combination therapies. So looking at gamma delta T cells, 1984 with the discovery, and coming now to 2018, where they're now easily available in clinics. There are CAR T cells which are being generated. And I'm not only fascinated with CARs, but there are also untreated gamma delta T cells, unmodified rather, not untreated, unmodified gamma delta T cells, which can also give a lot of advantage to patients. And there are clinical trials, especially in bone marrow transplant patients or in hematological malignancies, these gamma deltas can be used. So now there are a lot of companies. This is a very small list, much more than this, giving off-the-shelf treatment of gamma delta T cells for the treatment of cancer, used in different ways, used along with bispecific antibodies, along with cytokines, and as I said, engineered like CAR T cells. And I think this will be my last slide. Establishing a GMP facility at Attract was my dream. I managed to get funds for it, but it didn't happen during my tenure. But I'm happy that now Gaurav Narula and Rahul Purwar are taking it ahead at our center. It started at our center. There's a phase 1 clinical trials which was successfully done. But I hope they will someday start also with gamma delta T cells, or if Rishikesh is listening, Rishikesh can initiate this at the Immuno Act, because these are wonderful cells which can be exploited for immunotherapy. I will quickly browse through besides all this work, we did work on, and this was a collaboration, mind you, with Dr. Madhavi Indap, who was at DG Ruparal College. He was head of zoology and very enthusiastic. I've never seen a college teacher so enthusiastic in doing research. And she's a zoologist, so she used to collect these Turbobrunessus extract. And she came to my lab one day and said, can we do some immunological studies? And that's how it started. And we showed that we published in Bone that turbomethylal extract inhibits bone resorption through regulation of T cell functions. And all these papers were published from 2 students, Sachin Chaugule and Babita Balkrishnan, who worked in my lab for this part of the work. We have also done some work, and I would be failing in this if I don't mention Aparna Chaudhary, who did all the work on galactines in my lab. And we collaborated with Rajiv Kalreya because he had this galactine knockout mice and showed the role of galactines. Swati Patel in my lab, we studied how this is again contributing to the immune dysfunction, because the non-small cell lung carcinoma patients produce tons of IL-10, and we showed that how IL-10 regulates, how IL-10 production in the non-small cell lung cancer patients is regulated through the cyclooxygenase pathway. So a whole lot of use of cyclooxygenase inhibitors and looking at the regulation of IL-10. With Prakriti Tayalia from IIT Bombay, we started this work. Her student came to my lab to do the flow cytometry, and to use some animal work, and we did this work along with her. And we also participated in a consortium And there were several people, as you can see from the authors, in understanding the molecular variants of HPV-16 cervical cancer in Indian population. We also looked at region-wise, this was all DBT funded, region-wise distribution of high-risk human papilloma subtypes in India. I spoke about this industry-academia collaboration. We did another collaboration with the Bristol Myers Squibb in looking at colon cancer and looking at immune profiling in colon cancer. This part is not published yet. And another interesting story was the science initiative in Ayurveda, which was started by the Department of Science and Technology by Professor Valiathan and Dr. Professor Chidambaram. Dr. Valiathan wrote to me and said, because when he invited, I said to him that I don't know nothing about Ayurveda. And he said, we want you as an immunologist and your inputs as an immunologist. So we looked at immunologic and metabolic profiling of what happens to patients receiving panchakarma, that is basti. Basti in short means enema, and how this Ayurvedic basti, what happens to the immune scenario in these cases is a clinical trial. And with IIT, we collaborated in looking at the safety and bioactivity studies of Jasad Bhasma and its process intermediate in Swiss mice. And of course, all this along with my tenure at ATRAC as a scientist and the responsibility given to me as a director, which has been a very enriching experience. And so many of students, all my students are not listed here because this is the Gamma Delta team. There are so many other projects which were done. And my clinicians with whom I collaborated, had excellent support from all the clinicians from the Tata Memorial Center, who are not, some of them are my good friends, and collaborators from different institutes. And it's also from ATRAC, Pradeep Chaudhary, Murli Krishna, and from the ICTRC Center Wagholi, which is a beautiful Ayurvedic center, which we did, we are still doing in the ongoing study, a clinical trial with them, with these patient breast cancer and oral cancer patients. And all the funding agencies which have generously funded the research in the lab. And more so, this journey would not have been possible unless the support from my excellent team of scientific assistants, technicians, all are not there in the picture, but a wonderful batch of students, which I was lucky to have. Thank you, and thank you for your patient listening.
Thank you very much, Dr. Chibrunkar. An excellent talk and a testimony of how most focused and dedicated principal investigator, with the help of so many bright students, can handle a variety of aspects of a particular project and then take it to a situation where they can be applied in the human beings. Thank you very much indeed. An excellent lecture. And may I now ask the audience whether they would like to ask any questions to Dr. Chibrunkar? I'm sure she is happy to answer them.
Yeah.
It was a wonderful talk, Dr. Taruna. Nice to see all of you, and Dr. Sainis after a very long time. So thanks to for arranging such a wonderful talk. All the best.
Thank you, sir. Thank you. May I now invite Professor Shamal Roy? He had a query.
Can you hear me?
Yes.
Yes, sir.
Yes.
Shubha?
Yes, yes.
Okay. For my own knowledge, I wish to know that the gamma delta T cell that is coming out of the thymus, is it involved thymic selection process during the development, or it is independent of thymic selection process?
No, there is thymic selection, and in fact there is a notch which plays the important role and these are opposing roles. In mice, a notch dictates alpha beta lineage, and in humans it is the gamma delta lineage. So as I mentioned, the mouse situation and the human situation are very different with respect right from lineage discrimination to the antigen recognition.
Do we have anyone else who has a query?
Shubhda, is there any published work or information on whether any of these gamma delta T cells express other forbidden receptors which can activate autoimmunity?
Like, yeah,
We
they
know that in case of
told,
double negative alpha beta positive cells.
yes, yes. Like, you mean to say toll-like receptors, pattern recognition
Yeah.
receptors? They are there, yes. In fact, Bhairav Paleja from my lab, I didn't present his work, but he has done work on the toll-like receptors expressed on gamma delta T cells. And we've also looked at, the polysaccharides which activate gamma delta T cells to the pattern recognition receptors.
What are the antigens in the thymus for selection and all these phosphoconjugates, something like that?
No, but in mouse it is known, because in mouse, you have this H2 complex antigens, which are known, but in humans,
not so much clear. And for your CAR T cells, CAR gamma delta T cells, because we know that the CAR T cells is very effective for the liquid tumor, but not for the solid tumor.
Yes, it's
so
a challenge. Yeah, complete your question, then I'll answer. Yeah.
Same thing is also for the CAR gamma delta T cells as well.
Yes, as I showed also, most of the work on CAR with alpha betas is on lymphomas and leukemias. Solid tumors, the challenge for solid tumors is what is the antigen, because tumor-associated antigens, are difficult to define. But there is a lot of work going on mesothelin, on HER2 and other antigens which are expressed. The problem.
I can hear you, Subha.
Yes, there's work going on. The success stories, or so-called success with tumors. In solid tumors, there are challenges with persistence, with penetration of gamma delta T cells and exhaustion. So these are not overcome. But the paper which I showed flashed, it was in pancreatic cancers, and this is the first paper which has shown, especially in bone metastasis pancreatic cancer, a CAR T designed with gamma delta T cells.
So why there is less synapse formation with the alpha beta versus gamma delta T cells?
These are leukemic blasts. The synapse is, when we are talking, remember, these are the leukemic blasts. Okay. The adhesion molecules which are required are different between a leukemic gamma delta versus a leukemic alpha beta.
Okay. Thank you, Shubha.
Dr. Saini?
Is there anyone else?
Yeah, Dr. Saini, if you grant me permission, I would like to ask a question.
Sure, by all means.
Thank you. Ma'am, a fantastic presentation, and I see so many accolades in the chat box for you. And,
I can't see them. I can't see them.
But I have taken a screenshot for you.
Okay.
So I will pass on these to you. My question, being a reproductive biology person and immunology person, is that you said the BPP and the other agents that are coming, which are substrate or antigens for these gamma delta T cells, they are coming from the cholesterol pathway, right, ma'am?
Cholesterol metabolism pathway, the mevalonate pathway.
Yeah. So are there I don't know, that's not have been your, this has not have been your area, but my question was, are they known to be there at the feto-maternal interface?
Yes.
And because our steroid hormones are also coming from cholesterol, so are these gonadal steroid hormones, they have an impact on the gamma delta T cells?
No. See, the thing is different. The steroid, this pathway is important for the mevalonate pathway, is an obligate intracellular pathway for formation of vitamins, dolichols, alcohols, and everything. So that has nothing to do with the intermediate of that pathway, which is isopentenyl pyrophosphate, is expressed, and I would say strong connections, because of your chlamydial infections or any sort of infections would definitely have a impact on this pathway. But presence of gamma delta T cells in the trophoblast is known, and there are a lot of reports now looking at the HLA-G and HLA-E interactions and being immunosuppressive rather in the whole thing. Yeah.
And if you permit, sir, I had another question. Ma'am, as we have subtypes for various, T cell subsets, so in gamma delta T cell also, do we have some subsets? As you showed two of them, right? Based on the receptor chains, you showed two of them. But do we have something like Th1, Th2 based on the cytokines that they stimulate?
Which is the gamma deltas released, you mean to say? Defining the gamma deltas as Th2, Th1, gamma deltas are dominantly Th1 like, they secrete a lot of interferon gamma. IL-2 is something which they need, or they are like dependent on IL-2. So they secrete a lot of interferon gamma.
Because I did see that you showed in one of your slides that there were CD25 positive gamma delta T cells.
Yes.
Right? And so do we
This is an activation marker. So CD25 is a late activation marker, which is expressed on gamma delta T cells.
Okay. Because you said that IL-17 is also out of some of these is induced by gamma delta T cells, and they have a role in autoimmune diseases.
There's a differentiation of the Th17 phenotype lineage because of the hypoxic conditions in the tumors, which drives the Th17 phenotype. But yes, there are so many examples of this phenotype in SLE and psoriasis, where there is an outgrowth of this phenotype.
And ma'am, the fantastic difference that you showed in the T cell leukemias, the blast showed that if you have a particular gamma delta receptor, and then the patients actually do not need that kind of strong therapy. So is it being taken forward?
Very difficult. I'm sure they will, because Dr. Banawali is very interested. He knows this work very closely. He has worked with us. It's up to them now.
Sure. I can see KP.
If the chairman allows, can I ask a question?
Hello.
Yeah.
Hi, KP.
Hi. I was on the wrong connection. That's why I couldn't talk to you guys. Hi.
Hi. So nice to see you after a long time.
Yeah, nice to see you. That was a lovely lecture, Shubha.
Thank you.
I just wanted to ask you a question.
Yeah.
What are triple negative breast cancers?
Yeah. Triple negative breast cancers, because breast tumors express hormone receptors, estrogen receptor, progesterone receptor, and they are HER2, which is the EGFR family receptor. And these triple negatives are very difficult to treat, because see, when there are hormone receptors, you can use treatments like, if it's ER, you can use tamoxifen to block, to make the hormone unresponsive, do orectomies to remove the source of the hormone. So these are different modalities of treating those patients to remove, when they're hormone responsive tumors. But a triple, it's very difficult to treat.
Yeah.
And they're mostly aggressive tumors.
Yeah, and one of the reports that I just read, since you were going to talk, so I just read something about gamma delta cells, where treatment makes them susceptible for gamma delta cytotoxicity. So is that a repurposed drug which is now being used for these aggressive breast tumors?
No, it's not a repurposed drug. The bisphosphonates have been, they have been administered to patients. First, it started with treatment of osteoporosis, just to treat patients where, to treat them for osteoporosis. And now, because of their role in cancer, they're being used. So they're easily available, FDA-approved drugs.
Okay, thanks. Very good.
Thank you.
Nice to see you, Sinus.
Yeah, Uma wants to ask a question.
Good afternoon, ma'am. What a wonderful talk, and I'm really in awe of the work that has been done in your lab. Amazing. Thank you so much for sharing with us. My question was, ma'am, since you explained there are different functions and different ways that these cells can act, is there any hierarchy in their function that you think is going to be best utilized for therapy? For example, as an adjunct therapy, or in terms of the Th17 potential? is there any hierarchy in this function of
You're
these
talking
cells?
hierarchy between alpha, beta, gamma, delta, or gamma, delta? Hierarchy between what?
Just the gamma delta. Which ones? Which ones be more potential for using in therapeutic ways?
As I said, see, there are trials being done with unmodified gamma delta T cells, okay? And there are trials being done with engineered gamma delta T cells. None of them are standalone therapies. They are to be given in combination. So the adjunct therapies, and standard of care is always there. Standard of care treatment to the patient is always there. Because these are just emerging therapies, even for checkpoint inhibitors, today PD-1, PD-L1, we know that they are in combination, given in combination, or along with the standard of care therapy.
Thank you so much. Thank you. A lot of learning.
Anyone else? Oh, no more questions? Okay, once again, I thank you very much, Subha, for an excellent talk. So much educative for all of us, even if I was somewhat familiar with what your students were doing. Even then, there are so many new things that have been done subsequently. And on behalf of Indian Immunology Society, as well as on my personal behalf, I thank you for this excellent talk and wish you all the best. I'm sure you are going to continue with this work in some form or the other, or in some place or the other. And wish you all. And I also thank
some of the youngsters to take up gamma delta T cells and continue the story.
You know, sometime Taruna will tell you, Pramod Khandekar joined Sharmila Bapat in NCCS, Pune. So if you have a position, tell us, some of us may join. That's a good idea. I had once threatened Karnuri Rao that I will join him as a postdoc.
Yes, I remember.
We would love to hear you at the Golden Jubilee conference.
My God, I've kept it at an overlapping with my the IIS education committee. How will?
But this is 6, 7, 8. So you after the course, if you come here, a lot of students will be, very happy to hear you. So we really want you to be with us.
All of you, we will just present a formal vote of thanks, and I request my colleagues.
I want to thank Indian Immunology Society for giving me the opportunity to chair this talk. Okay,
thank you so much. Yeah, we'll present a formal vote of thanks to our chairperson and speaker, and my colleague Dr. Rupesh Srivastava from All India Institute of Medical Sciences, Delhi, would do us the honors. Thank you, Rupesh.
Yeah. Thank you, ma'am. So, very good evening to all of you. So, on behalf of the Executive Committee of the Indian Immunology Society, we have very immense gratitude, and we express sincerely and wholehearted thanks to our eminent speaker, Professor Subhda Chiplongar, ma'am. She has been a former director at ACTREC TMC, and she's also president of Immuno-Oncology Society of India, along with being the member of IIS Education Committee, for delivering this prestigious 6th IIS Golden Jubilee lecture and sharing her critical information on the role of gamma delta T cells in cancer, a topic which is very much synonymous with her name. Ma'am, it was really an exhaustive and interesting journey for us, and we are really honored by your presence and sharing of your rich experience in the field of basic tumor immunology, with the strategies that have been involved in immune evasion of tumors and translation of this knowledge to newer immunotherapies. And one more thing I would just like to add here, that you have been also by Dr. Chiplongar had been my Viva examiner for my PhD Viva at NCCS under Dr. Vani, and it was in somewhere in July 2011. Ma'am, hope you remember me.
Yes,
it was really an honor to.
And I'm very happy that you are taking the work ahead so nicely. Very good, interesting.
Thank you, ma'am. Thank you, and I was really grateful to ma'am for basically when she called me today in the afternoon. That I have to give this vote of thanks, and I mentioned her that this was the story, so she was also okay. You can mention this. So thank you for that. And also I express my heartiest thanks to the chairperson of this event, Professor Krishna B. Sanis. He has been a former director of Biomedical Group BARC, for having accepted and orchestrated this event in a very scientific way. And we again thank all the participants across the country for their overwhelming response. And thank you all. And last but not the least, we thank the IT team at ICMR NIRRCH. Without the help and support of them, this event was not going to be a success, which is we have already seen that. And thank you all, one and all, and a very good evening to all of you again.
Thank you everybody. Thank you, Taruna, and all the EC members.
Now carry on. Thank you, Rupesh. We can carry on with our non-formal discussions, please. This is a forum meant for this.
Oh, I can see Savita. Hi. Yeah.
What else you are doing other than science?
What else? In the sense, I'm doing science.
Apart from science.
Apart from science,
Hi, Krishna.
I've joined two music circles. Shamar, you're not listening to my reply.
Pardon me?
You asked me what you're doing besides science.
Yeah, that's the thing.
My reply. I joined music circles. I enjoy listening to music. It's a very unique one. Those who are interested, if you write to me, I can tell you. It's an interactive music sessions.
That's good.
It's called Nostalgia. If you can Google it, you can become members of Nostalgia. I go for stand-up comedies. I go for theater, cinema, everything.
Very good.
But I'm still very much into work. I must say.
What's the name of the site?
Nostalgia.
Nostalgia. Hi, Shama.
Jukebox.
Hi, Krishna. How are you? Long time.
Good. Nice to see you after a long time.
I've seen KP after so many years. But KP, you have no change. Absolutely the same. No change. Yeah.
Lost a lot of hair.
No.
Incidentally, now Shubhada is the independent director on Origin.
Yeah, I've joined Origin Oncology, which is Dr. Reddy's.
Origin Oncology.
Oh, lovely.
Yeah.
I joined in the sense they invited me, so I joined. It's interesting. They're doing good work.
Must be a lot of work, huh?
Yeah, shuttling between Hyderabad and Bangalore.
Wow.
It was nice to hear again, Shubda, from you.
Yeah.
Nice to see Dr. Prasad after a long time.
We can't see you.
We can't see you, Sripad.
Yeah.
Video is off.
Bala, it was nice. It's nice to see you after a long time. Sripad here.
Yes.
We can hear you, but we can't see you.
Kaustav is there. Kaustav is not saying a
Kaustav is there. Hi, Kaustav.
Yeah.
Hi, Savita.
Can I take this opportunity to once again remind you all of the Golden Jubilee conference that is happening on October 1st week, October 6th to 8th. And many of you will be receiving the invitations from us for a talk. So I'll request you all to please accept that and encourage your students
Send us a
and
program.
other members of IIS to join.
Where will it be?
Filling up the program and sending the letters. We are getting started sending invitations.
Where will it be?
The dates are 6 to 8. So October 5 is the inaugural. Then we will have a quiz and some activities for students. And 6, 7, 8 of October at AIMS at the Jawaharlal Auditorium. We'll have the conference. It's only a few months and very soon you all will receive all the details and invitations. My humble request to all of you to join and make sure that we have a good Golden Jubilee celebrations of the society.
The Jubilee conference was in Bombay and Dr. Chiprunkar and I were looking at it at that time.
That was a wonderful visit.
Shubha, do you remember?
Yes. We really enjoyed that conference.
Yes.
I must tell this. We have time, little time to.
Yeah, sure.
KP, you were there, no? Conference in Mumbai.
I was there.
And there was a strike declared just one day before, hotel strike.
Yes.
And Dr. Sinus and Jayashree served dinner. All those who were coming that night.
Rajni Rani and I think Salunke.
Yes. Oh, it was
KP, you are in Delhi or elsewhere?
I'm in Mysore.
Oh, Mysore. You're not Basically settled in Mysore.
Yes.
That's your hometown.
Yeah. That's my wife's hometown.
So what you are doing there?
At the moment, nothing. I was involved with a local medical college here.
Okay.
Now I'm doing, that term is over. Now
some teaching?
I'm unemployed now.
Okay. Good. Very good.
All of us are.
Shamal, you went for the MIF? Who attended MIF?
Nobody is inviting nowadays anymore. So kind of old timer.
I wasn't well at that time.
Say, Dr. Shamal.
That was for people below 45 years.
Kind of
No
old
more
timer.
below
But
45.
I heard, I met Anna, and Anna said they all attended. Satyajit, Anna, Vinita, all attended.
Okay. So next time.
What I was worried about is that bus journey from Delhi to Jim Corbett Park. That was just too much.
You have to talk about some science. We are not doing science anymore. So not much to data, and so now Shubha is still active, and all the glorious data Shubha is presenting. I envy Shubha that so many beautiful things is continuing.
Absolutely.
But I don't have any data to present, so therefore kind of staying away from MIF.
But I really miss the lab. I miss the lab.
Yeah. Miss the lab.
We all, of course.
Yeah.
So I guess you have to look for new avenues.
Me?
No, I'm saying if you miss the lab, you have to look.
I should do some advertisement for myself. So my book of poem will be published sometime next year. So I try to do some advertisement. So you buy a copy so that I get some royalty.
What's the title of the book, Shamal?
Are they in Bengali or English?
100 golden lips.
Wow.
Published from will be published from London. Some company interested.
But in Bangla or English?
I do some advertisement for my book. So buy one. Don't lend it to your friend or anybody else.
Shamal, you had written something on art and this, I remember.
Yeah, it's something like that. So
Yeah,
whatever
very nice, very nicely
coming
done.
in my mind. So I started writing. So just doing some advertisement. So buy one so that I get some royalty.
Sure. Let us know.
But don't give it to others. So whatever you buy, keep it in yourself. Okay.
Hey, Shamal,
Yeah, hello.
Are there some poems on the time you spent at Harvard Square?
Oh yeah. Those things are there too. Dr.
Shamal Roy, we are sending you the invitation for Golden Jubilee. So we would like to share your experience.
Yeah.
Even if there's no new data, but your experience
will help. The speaker next time?
Dataless speaker.
Who will be talking about next time?
Some of us should be dataless speakers.
Dataless.
So yeah,
we can have.
But Shamal gives a wonderful talk on art and science.
Yeah.
At least I heard it. Very nice.
Lot of
youngsters will. The analogies he has drawn.
I'll be happy to give one.
Okay, so for the Golden Jubilee, we'll keep that, ma'am.
Yeah. I strongly recommend. Ask him to give a talk.
Yes, sir. Please. So your commitment now only. So
I'll be happy to give one. Okay.
Yeah, that will be great. Thank you so much for agreeing.
Honored. Thank you.
It will be our pleasure and really honored. Okay. That will be nice. So we will update you all very soon about the program.
Thank you.
Thank you.
Stay well and connected.
Yeah.
Thank you very much.
Thank you, Dr. Balakrishnan. He's the only one who's.
We look forward to see you all in the next one.
Yes.
I'm seeing Dr. Balakrishnan for the first time.
Who's the next speaker?
Dr. Balakrishnan has convinced Dr. Bala. Can you please let us know what is the date decided for the next one? The next Golden Jubilee seminar?
Oh, he logged out.
No, you have to unmute. You have to unmute.
Aruna, I'm again logging out. I'm again going back to the other meeting. Sorry.
Accepted on 19th or 20th.
Bye, ma'am.
Bye.
Bye, sir.
June. So the next.
So who is the speaker? Next speaker?
Dr. Muthukarupan, you said, no, sir?
Great. Wow.
Yeah.
Again, we'll be meeting Dr. Muthukarupan after a very long time.
Yeah.
Yes, 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 yes. It is, we are
Okay,
grateful.
thank you.
Yeah, thank you. Thank you, everyone. Thank you, Shamil sir.
Thank you all. Wonderful
Thank
meeting.
you.
Thank you, Ma.
Thank you.
All the best for the conference.
All the best.
Golden
All the
Jubilee
best for the
conference.
Sir, we need your support. You have to be with us.
Okay.
Yes.
But
Yeah.
only the last, only on 6th. Let's see.
So 6th, 7th, and 8th is not an overlapping, so you will be able to join us.
Okay.
Yeah.
Thank you, everyone.
Thank
Bye.
you.
Okay.
Bye-bye.
Bye.